Hi, my name is Brooke, and this is the Science Spectrum's Female Scientist Fridays. This week's female scientist is Rosalind Franklin.
Rosalind Franklin was a famous female chemist and physicist whose research with X-rays would be important in discovering the double helix structure of DNA.
DNA is a really tiny instruction manual for our cells, which are the building blocks of our body. DNA tells our cells what to look like and can decide whether one person has brown hair or blonde hair. By figuring out what DNA looks like, scientists can learn more about diseases and how they affect our body through our DNA.
Rosalind Franklin was born in 1920 in London, England. She studied chemistry and physics at the Newnham Women's College at the University of Cambridge.
After receiving her PhD, she moved to Paris to work in a research lab. When she was in Paris, she helped pioneer the field of X-ray crystallography, which uses X-rays to create pictures of really small things and to help us find the structure of objects that we can't see without the help of tools like microscopes. In 1951, she moved back to England to work at King's College. Rosalind worked with another scientist named Maurice Wilkins to find the shape of DNA, but they did not get along and they chose to work by themselves. In 1952, Rosalind Franklin took the now famous photo 51 of the structure of DNA.
This photo was taken in Rosalind's lab, and while it doesn't look like much, it was the most important photo that could help discover the structure of DNA, and Rosalind wasn't the only person who thought that.
Maurice Wilkins, her colleague, saw her photo, and without her permission, he showed it to two other scientists who were also trying to find the structure of DNA, James Watson and Francis Crick.
Watson and Crick then used Rosalind's photo in their own research to determine that the structure of DNA was indeed a double helix, just as Rosalind and her team were coming to the same conclusions. The double helix shape essentially looks like a ladder that's been twisted around.
Watson and Crick used the photo to write about the structure of DNA and publish their findings in 1953. They did not acknowledge Rosalind's help in determining the structure of DNA, and she was not given credit in their paper. Watson and Crick are now credited with being the ones who found the structure, and her name is often never mentioned in this argument. At the time, women weren't treated fairly in their science jobs. Rosalind said that she wasn't paid the same as the men that she worked with, and she wasn't even allowed to eat in the same cafeteria as her male coworkers. But she didn't let this unfairness stop her. She continued her research, and in fact, she was friends with Watson and Crick. It is debated today whether or not Rosalind knew that Watson and Crick had seen her image without her permission, but even if she did know, she never talked about it. After 1953, Rosalind continued her research in X-rays and learned more about the structure of viruses and coal. Sadly, Rosalind died in 1957 from ovarian cancer.
Her photo 51 would prove very important to the study of DNA after her death, but she was rarely acknowledged for her contribution.
In 1962, Watson, Crick, and Maurice Wilkins, who had showed her photo to the pair, won a Nobel Prize for their work in finding the structure of DNA. Rosalind could not be eligible for this award since it was not given to scientists after they died, and it is uncertain of whether it would have been given to her anyways if she had still been alive. While Rosalind's contribution was not acknowledged during her lifetime, her name is now remembered and her work is appreciated more than it ever was. James Watson later said how important her photo was to the discovery of the double helix structure, and he even admitted that the discovery would not have been possible without her. Rosalind Franklin is now getting the credit that she deserves, and modern scientists can use her work to learn more about microbiology. You can learn more about the structure of atoms at the Science Spectrum.